So I'm Sam Landis. I am the treasurer and membership coordinator of the Mushroom Club of Georgia. And we always like to remind,
whoops, and go ahead, dear. What else?
we always like to remind people it's the Mushroom Club of Georgia. It's not the Georgia Mushroom Club.
Right. We don't have a catchy name like SCUM, the South Carolina Upstate Mycological Society. They were brilliant. Or COMA, Connecticut Mycological Association. But yeah, so I'm the Cornelia Cho, I'm the president, and then I think
Jeanette's down there.
Yeah, I thought I saw Jeanette. Would you like to introduce yourself?
Hi, I'm Jeanette, and I'm the secretary.
Yes. So thank you, everybody, for all the work you do.
So you want to talk any other news, dear?
What did you have in mind?
Club business?
Yes, I am excited.
Uh-oh.
I don't see Brad on here, but Brad Beatles is
You've all frozen on my screen.
not on my screen.
Yeah, it's just you who's frozen right now.
Yep. So we have a newer member. His name is Brad Beatles, and he has been growing oyster mushrooms on English ivy and privet.
Wow.
And he's taken the trimmings that have been collected from parks that are trying to clear the plants out, dried them, then put them through his home wood chipper, and then pasteurize them and using plastic buckets, 5-gallon buckets, has been able to get really beautiful fruitings of oyster mushrooms. And so sometime in probably February, we're going to be having a workshop. And what's even more exciting, he's working with Joe Sexton at Clyde Shepherd Park. And Joe has actually collected an oyster mushroom that occurs in the park already and is cloning it. So he'll get a local mushroom sample to eat the local invasive vegetation.
That's wonderful.
So yeah, it is cool. I see Karen saying it's cool. So keep an eye out for that. That is something we're going to be doing.
Cornelia?
Yes.
I need to make you host because I dropped out for like the 4th time.
Yeah, go ahead.
So you're host, and then if you'll make me a co-host, I can take care of people coming in.
Okay. We'll do that. Okay, there you go.
Excellent. And can I say something?
Yes.
Okay. So being inspired by Sidnee, we went and got our own UV blacklight flashlights and been seeing some fun stuff. But since most of our walks are at parks, it's like finding a place where we could go at night is challenging. But our delightful, wonderful member, Sandy Aceto, who is in charge of a few Gwinnett parks. She's the reason we're actually at McDaniel Farm Park for the potluck. So she's in charge at the Gwinnett Heritage and Environmental Center. And they have trails. And so she has agreed that we can do a nighttime walk out there. So we're going to do our first MCG nighttime walk. And Cornelia, you're all frozen again, so I'm going to drop out. So I can't tell if anyone's hearing me at all.
We could hear that.
We could hear all of that, dear.
If you just turn off your video, it might help.
Yeah. Michael Fletcher, do you mind helping out with admitting people? Because you've done that before.
I can't because I'm not on my laptop tonight. I'm on a phone.
Okay.
I don't think it'll work with a phone.
Would somebody else like to help
Hell
out?
said she could, if you want to make them a
Okay.
co-host.
Right. Okay.
Because it's problematic with me dropping out.
Yes, it is. Okay, we just did that. All right,
Yay.
great.
Thanks.
Okay, any other announcements, dear?
Well, did people hear? I mean, because you all froze, so I don't know if you could still hear me talking.
We heard everything you said.
Okay, great. So we're gonna do our first club nighttime walk sometime, probably in January.
That's great to hear. I think it's probably the first club I've heard of doing like formal night walks.
Yay.
It's a step in the right direction. It's been interesting seeing it like gain popularity, just as kind of a new category.
Cool. Well, thank you for inspiring us.
And I don't know how many scorpions will be out in January, but we'll see.
Scorpions in January, probably not.
If they're there, you'll see them.
Okay, and hi, Karen, glad you're on. So sweetie, do you want to introduce Sidney? And he's already made a co-host, so he just needs to pull up his stuff when you are ready. So
Okay. Well, we're very excited to have Sidnee Ober-Singleton here tonight with us. And I think one of the most special things about you, Sidney, is that you're, you know, kind of self-raised in a lot of ways in this particular topic. Just Sidney got introduced to mycoremediation, the concept of using mushrooms to clean up the environment at Lane Community College, getting involved in a project there, and it just set him on fire. And
Yeah.
I know a few people besides you who love talking about mushrooms as much as you do, but it's been really great to get to spend time with you. We were together at the International Fungi and Fiber Symposium recently in Port Townsend, Washington. And I know you and I were up at least until 1, and I know you were up with other people till even later in the morning talking about mushrooms. So
It's impossible around so many people. Like,
yeah, that was an amazing experience. But so Sidney has definitely taken Gary Linkoff's advice to make your life be all about mushrooms and to pursue what interests you. And I love how you found it and discovered all these interesting, strange, unusual uses for mushrooms and ways to look at mushrooms. For instance, you know, we were talking about using fluorescent light, and there's just, Sidney has a number of topics that we could choose from to present to the club, and we basically picked the ones we thought were the most interesting to us. So the fluorescent fungi, and then also the predatory fungi, the fungi that hunt and eat things to get more nutrients. So that's what he'll be talking about. But we also asked him to include a special piece that he has. He has a little movie about dyeing things blue using a specific mushroom. So I'm really excited to welcome here Sidnee Ober-Singleton and have him be our speaker tonight. Thank you so much for joining us, Sidney.
of course, I'm really glad for the opportunity. And it's like, you know, a lot to talk about. These are definitely two of my favorite topics. And then to be able to throw in that video at the end with some explanation, I think people will really like that. But yeah, sort of two topics that are not talked about as much as I think they should be. Both are kind of really gaining popularity and steam. Like in particular, the predatory fungi. I sort of discuss a method used for exploring those. And I've recently been hearing about people actually picking it up and getting hands-on with it, which is something that like amateur level people haven't been doing since the 70s, I think. So it's really good to see stuff like that. But I'm gonna kind of like, I normally give these talks just stand alone, and they usually run about an hour. I've kind of like chopped them down a little bit so we have time for both. We'll see if Cornelia and Sam could kind of pay attention to the pacing of things. If I drag one out way too long, please just give me a reminder so I can hustle things. And I think I'll probably like feel free to put questions in the chat. But I think what we're probably going to do just to manage time a little better is save most of the questions for the end. So I'll be doing predatory fungi, then fluorescent fungi, and then just a bit after that on the dying. If there's something crazy important that a lot of people are asking, then yeah, please shout that out. But yeah, I think I'll probably go ahead and get started if everybody's ready.
Right, and we often ask people to turn off their video because that helps the speaker's bandwidth and it helps things come across better. So thanks for shutting off your video.
Sweet. I'll go ahead and share my screen.
But feel free to put stuff in the chat.
All right. Is that looking good? Everybody can see it?
Beautiful.
Looks great.
Sweet. Yeah. And if anybody's having trouble hearing me, let me know. I think my mic and everything is all adjusted. But so this first talk is on predatory fungi, probably one of the least discussed ecological roles that fungi have. You know, we really often talk about 3 ecological roles as though it sort of like encompasses everything fungi do. You know, the common ones being like saprobic fungi, which consume dead or dying material, other sources of like carbon they don't have to fight over. There's mycorrhizal, which is a huge amount of the most commonly foraged mushrooms out there. These are fungi that form associations with tree roots. And then there's parasitic fungi, things like honey mushrooms, armillaria, or lion's mane, or chicken of the woods, shrimp of the woods are all good examples of mushrooms that are parasites either on other mushrooms or trees. But that parasitic category gets really messy really fast when you look into the habits, especially when it comes to how they eat microanimals or, you know, different types of moving organisms. And so it kind of gets, I guess I'll go into that gray area a bit more. You know, for animals, predation is clear cut. When we think of a predator, we think of something like a shark, a hawk, or a lion, something that actively moves around in order to pursue its prey, something that has very specialized anatomy in order to capture its prey. The prey are also generally things capable of moving. Then when we remove the movement from the prey, predator is often a label that stops getting used as frequently. So when you look at, you know, things that graze or filter feed on actual animals like coral or plankton, or even things that eat bugs, predator often kind of like starts to vanish. When you remove the movement from the organism doing the hunting, which pretty much all fungi are, all terrestrial fungi are primarily immotile, completely immotile. There's a few stages that have flagella and moving zoospores, but they're not eating in that stage. And so when you have a predator that's not doing any movement whatsoever, it gets even dicier. And the concept of what constitutes a predator in fungi gets real messy real quick. So this is probably the most agreed upon, simplest definition of a predatory iconic fungus. And so this is Arthrobotrys. Pretty much the whole group uses nematode traps in some form or another. Now, nematodes are close to microscopic, sometimes microscopic animals. They're similar to worms in appearance and movement, but their ecological roles are all over the place. Their biomass in all of Earth's ecosystems are absolutely insane. A lot of researchers who study nematodes don't even necessarily have to leave their house to find specimens. I don't remember who it was, but there was this great description where if you were to remove everything from the Earth, like just get rid of the earth, all the items, animals, etc. on it, except for nematodes, you'd still be able to see everything because there'd be a white crawling film on every single surface. Like nematodes have made themselves room in every single habitat on Earth, eating practically everything. Some of them get multiple feet long in our digestive tracts. Others are plant pathogens or fungal pathogens. They're all over the place, really. And so probably the most popular predatory strategies among fungi are different ways to capture, kill, and eat nematodes, which are moving animals with defenses of their own. In general, they're eating ones that are pretty small, but some of the strategies out there have actually developed to be able to catch ones far bigger than things like these snares. So these Arthrobotrys snares were actually the very first predatory method described in fungi. It was actually a researcher named Zoff in the late 1700s who found Arthrobotrys oligospora capturing and consuming nematodes. It's a very distinct method. These snares are really widely distributed throughout predatory fungi. Arthrobotrys has a remarkably well-adapted one. I'll go into the mechanism on that in just a second, but I would love to show off this photo. This snare method actually goes back well before many other animals were even alive or moving. So this is actually an amber fossil from 100 million years ago showing a non-constricting nematode trap producing fungus. This was about 100 million years ago, but genetic evidence suggests that they probably developed about 400 million years ago, which is actually right after nematodes first really evolved on terrestrial ecosystems. This method is ancient, very effective, and they basically started catching and eating what were some of the most dominant animals at the time, right as soon as they evolved. I think that's absolutely wild. There's really two categories of these snares. There's constricting and non-constricting. So this would be an example of a non-constricting snare. It acts almost more like a collar. This is Arthrobotrys. So these are active snares. So they basically a colony will detect the presence of nematodes, generally by what we could consider smell. It'll pick up different compounds, pheromones, traces that nematodes produce when they're in the area. Or for example, if a nematode tries to eat them, they'll start producing snares in response. And these are basically 3-cell loops that are being put out there on the periphery of the colony, sort of right littering the surface where nematodes tend to crawl around and explore. When a nematode disturbs this snare, when it touches the interior, sort of through a combo of mechanical and chemical detection, these 3 cells actually rapidly inflate. So they go from this open loop to changes in osmotic pressure that causes a sudden influx of water. They inflate, and all 3 press in towards the center to constrict actively around an unfortunate nematode that's poked its head in. Usually this constriction is violent enough that it'll practically break the nematode when it happens. They don't exactly have necks, but it basically snaps them and kills them on the spot. After the nematode's been captured, whether it's alive or not, a 4th cell will poke out from that snare, punch in like a peg into the nematode's body, and begin eating it from the inside out. And that peg actually first appears within an hour of the nematode having been captured. It's basically right away they start consuming the prey that they've caught, whether it's still thrashing around or not. The nematode can't do much at that point. And then these non-constricting snares actually seems like they might be worse because they don't actually tend to kill nematodes right away when they're caught. These sort of go for a different angle. So oftentimes these non-constricting snares are much bigger, wider, and in the constricting snares, or just any of these, if you have a nematode that's too big and gets itself on one of these, it'll just break away. It's actually large enough to damage the mycelium and break it off. Now, with the constricting snares, they're deliberately sized down to where they'll only catch nematodes that are small enough to be restrained. With these larger ones, they actually encourage that break. So a larger nematode will poke its head in, thrash around and break it off, and it'll be wearing that snare like a collar, basically, around its body. It will think it's fine, continue foraging, mating, etc. It'll travel away from the original colony before that peg appears, punches in, kills and eats the nematode, and assimilates it into a new colony. So despite the fact that the original colony wasn't able to hold on to it and take advantage of those resources for itself, this nematode will end up cloning it and carrying it off, acting as a vector for an entire new colony to form. It'll eat that nematode and produce more snares with the nutrients it gets. So it sort of gets a meal and dispersal from nematodes too large for other species to capture. And then after Zopf described this Arthrobotrys snare, it sort of blew up. A lot of different researchers, a lot of different mycologists, and even farmers started looking into this as a method to control nematodes, or just in general, because this was an area of fungal diversity nobody had really looked at. So I wanted to go through some of the bigger types of traps that we've found going up until now, before going a bit more into the history of all this, because there's some pretty cool stories. So this is one of the other really common methods. These are adhesive nets produced by a wide range of species. It's very similar to the snares, except they tend to be messier. You'll have multiple combined into one, and they're often sticky. They're either sticky or poisonous, and they're virtually always baited. So some of these fungi will actually produce either nematode mating pheromones to bring in nematodes thinking they found a mate, or there's feeding pheromones that nematodes will release as well. When they find a meal, they'll put off pheromones to basically congregate. And so these will produce one of the two or both in order to actually lure nematodes into these traps. Down here on the left, you can actually see a nematode that's fully involved, just totally captured. And sort of to put it into perspective, you can see the tiny diameter of this mycelium in the back. Like, these are thin cells. This is not very much biomass in one place. If you were to total up the weight of all that mycelium in one spot versus the weight and nutrients available in the nematode it successfully captured, the trade-off is insane. Their strategies for capturing these as a source of nutrition is extremely successful, which is why we've been seeing it for probably about 400 million years. This is actually a really cool one. So these are stephanocysts produced by Hyphoderma and a few Corticiales fungi. For a long time, we would see these on the surface of crust fungi fruit bodies, and we classified them as a weird type of cystidia. We didn't actually know what they were for, but they were called stephanocysts. We knew about them for a long time before realizing what their actual purpose was, which was a new form of nematode, not exactly a trap, but a way to colonize and consume nematodes. So these have usually one baited end and a ring of hooks going around the edge. A nematode simply has to brush by one of these. After it gets impaled, the spikes will inflate inside the nematode's cuticle or skin, basically, to adhere itself. And then down here, you can actually see, so this is a stephanocyst, and then all of these sort of blebs are mycelium germinating out. Because they can't necessarily punch in through the side, they'll actually grow their first hypha, their first cell, into the mouth or rear end of the nematode to gain access to the interior where it can start digesting it. But it was really cool to see when they actually finally realized that these were a nematode adapted method of dispersal and attack. this is another really cool one, and this ties into something I'll be talking about later. So these are called toxocysts. They're basically a droplet of poison contained within a very, very thin membrane. It's like a super thin walled toxin-filled cell. These are actually produced by oyster mushrooms, Pleurotus, when they're in nutrient-deprived conditions. So like wood, for example, the logs they often colonize, if that log is not touching the soil, it's extremely deprived of nitrogen. They'll do whatever they can to start getting nitrogen, which is basically proteins. That's the main way fungi get nitrogen. So it'll start producing these toxocysts all over its mycelium. They're basically almost like little mines. So the membrane is thin enough where any contact by a nematode will break that thin membrane. It'll wick out to sort of coat the surface of the nematode and paralyze it or kill it right at that spot, almost immediately after contact. It's a little horrifying, but as that paralyzed nematode is sitting there, surrounding mycelia will again seek out the mouth of the nematode. So this is one of those toxocysts up close. The membrane is super thin, so you almost can't see this edge here, but it's basically a droplet sticking off the edge of the mycelium. And then here you have a nematode that's been captured and all of this mycelium radiating into its mouth. And then this is later after full digestion. This one doesn't really show up very well, but it's you can see an extremely large nematode relative to the size of the mycelium. And then right here, this little dot is a single toxocyst. It touches that, the toxocyst releases its droplet, which is barely detectable in this image, but it's lost its droplet here. And then that nematode will basically freeze and die right where it sat. It's a really common misconception that Pleurotus also produce snares. I've heard that a lot. I think because Arthrobotrys with the snares is the most well known, and then people hear Pleurotus also ate nematodes, that they sort of conflated it. Pleurotus only produced these toxocysts. And there's been some really interesting research on the toxins they produce that actually target a nematode's nervous system. Some like really remarkably potent and well adapted poisons there. These are fun. So they were sort of affectionately called spiky balls. They're very, very sharp, almost Velcro-like looking asexual spores produced by Caprinus comatus, what people call shaggy manes, one of the inky cat mushrooms. This one is something, despite the fact we've like grown and known about Caprinus for a long time, this was a really recent discovery relative to how long we've known about it. We didn't realize until we put it on a nutrient poor medium that it actually produced these structures. It basically wasn't until it got deprived of nitrogen that it showed it had this ability for gathering more nitrogen by killing and eating nematodes. Basically, a nematode would contact one of these and tear itself open. It would just sort of gore itself and sit there as kind of a mess for the mycelium to sop up and digest. But it was really interesting how this was not caught for such a long time, similar to the stephanocysts. We've been looking directly at it for a long time without actually knowing what was going on. It wasn't until we deliberately deprived it of nutrients that we actually saw this. And it's very, very likely that there are myriad undiscovered strategies for eating nematodes, killing nematodes that we haven't even spotted yet, because this isn't something that's commonly done in like routine cultivation conditions. There was a particular guy who sort of pioneered methods for doing this that I'll get to in just a few slides. This is another really cool one. So these are called acanthocytes, basically spike cells. They're actually produced by the garden giant, Stropharia rugoso annulata, the wine cap, garden giant, king Stropharia. There's a range of different names for it. And it'll basically lay these out by the thousand, similar to the Pleurotus toxicis and the snares. It'll just coat entire surfaces in these jagged, gnarly cells, and nematodes will both be impaled physically and poisoned. So this photo actually was on the cover of Mycologia. This is a SCM image of an SRA acanthocyte showing these grooves and pores in it. Now, so far as I know, we haven't confirmed an active injection method, but these things are venomous. It works exactly like a snake's fang in some way to actually envenomate any nematode. If it didn't get a fatal wound just from touching one of these, it's still going to come away poisoned and die near enough that it can be consumed by the mycelium. I think it's interesting that it's garden giants that do this. It really just shows another way that they're so incredibly versatile. Really, you can feed SRA practically anything. But this is also one where you can see this yourself. If you start an SRA patch, if it ever gets a little more deprived of nitrogen, you can almost observe this as sort of a crystalline looking glint. Like they're very small, obviously, but you can see it as sort of a frosty looking glint on like wood chips just below the surface. It's not hard to find, or on agar too, you can see this. And then these are, it's definitely pushing it, but I feel like I really have to mention these. So these are actually produced by an oomycete. What we did consider fungi for a while, what we called water molds. These are actually closer to protozoans. They're no longer in the fungal kingdom. When we discovered these mechanisms, they were still considered fungi, but I feel like there's no way I can't mention these. So these are called gun cells. They're produced by Haptoglossa. They're insane. So if you're familiar with the way that the stinging cells on cnidarians, like jellyfish and anemone work, this is extremely similar. So a single zoospore will touch down, adhere itself to the surface. So you'll have an adhesive pad here. And the first thing it doesn't put out pseudomycelium. It's not directly trying to infect anything. It constructs this elaborate mechanism. So you actually have in here basically a dart, like a full-blown sharp pointy dart. You have a cord attached to it. You have a muzzle with an opening and a cap, and then you have this expanding vacuole back here. So after this is formed, the vacuole will swell and swell and swell and build a huge amount of pressure. Now what happens is when a micro animal comes in contact with this, now these are actually non-species specific. So like the snares can really only go for nematodes. These are actually effective against rotifers, what people call tardigrades or water bears, the cute little crawly things. They're effective against, rotifers and tardigrades. Rotifers are these little sort of, I don't know exactly how to describe them. They're another little micro animal that filter feeds and is also ubiquitous everywhere. Some of them almost get about 1 millimeter long, but they're overall pretty small. As well as nematodes. Some of these can even target things like amoeba or other protozoans. When something touches the muzzle, it suddenly like ejects, basically. It's been a little debated, but there is a ring around the muzzle, so sort of this cap right here, that's a designated break point. It'll snap, and all that pressure forces this dart out the front. It impales the organism that ran into it. The dart keeps it from backing out. And then this sort of basal zone here will actually inflate inside of the snagged prey. To tie it down, conveniently, the entire cell is glued down to a substrate. So it'll effectively harpoon and then tie down its prey and then colonize it from that embedded dart. There's an absolutely wild mechanism that I couldn't not mention, even though it's sort of fungal adjacent. So far as I know, it is only Haptoglossa, the genus Haptoglossa, that produces these. But amazing mechanism, truly. And now this is where I'll start going a bit into the history and sort of some of the how fungal predation actually helped form some of our modern families in terms of taxonomy. So after Zopp published his result with Arthrobotrys, that it had like very well adapted predation methods for nematodes, other people just like the field exploded because a lot of nematode, a lot of plant pathogens at the time that were resulting in huge crop losses, human pathogenic nematodes in South America, Africa, parts of the Middle East. Like nematodes are a humongous issue for humanity overall. They cause a lot of losses. So once Zopp described fungi very effectively hunting nematodes, some of these isolates kill over 99% of any nematodes present on their test plates. Very efficient mechanisms. Researchers just sort of dove into it, not only to discover new species, because countless ones had never been described. This is early 1800s, keep in mind, like late 1700s, early 1800s. So we really had not found a lot yet. We were just getting into it. Tons of researchers were looking for these. There was one researcher in particular, Charles Drechsler, who really got this down. Starting in about 1930, he began publishing outrageous quantities of new species, of descriptions, of applications for these in agriculture. He does absolutely phenomenal hand illustrations. He just kind of blew it up. He was an incredibly prolific researcher who published species for basically 50 years straight up until his death. He has entire genera named after him. He has countless species named after him. He was a huge mycological name at the time. Now, sort of important going back, this is about 1930, 1940, all of the predatory fungi that we had found had been Ascomyces. So there's really like the three main groups of fungi that we usually talk about, like the Zygomycetes, Ascomycetes, and Basidiomycetes. Ascomycetes are things like morels, cup fungi, etc. Don't have gills, usually on the weirder side. Then there's Basidiomycetes, which are like the gilled fungi, the common ones we usually go for. Probably the majority of the mushrooms you see out in the woods at a given time. And then Zygomycetes are a little more moldy. But up until that time, everything described had either been an Ascomycete or a Zygomycete. We didn't know that any Basidiomycetes had this strategy. One day, while using a method he had pioneered, which basically consisted of water agar plates. So water agar is a petri dish with agar and no nutrients whatsoever. It's basically a blank slate. It doesn't offer anything to what's growing in it. He would take a water agar plate and place soil samples or leaf litter in the center of it. And then because that soil or that leaf litter was the only nutrient source available and he wasn't sterilizing it, it was an intact soil eco sample, you'd have this entire little mini ecosystem developing in this 2D environment where all the organisms would be forced to interact with each other to gain nutrients, because the agar wasn't providing anything but a platform. It was just a way to sort of analyze this little microcosm. And so this is a method he was using when he found the first Basidiomycete that actually preyed on other organisms. He named it Nematoctonus, which roughly means nematode murderer or nematode killer, because it was so efficient. Unlike the others, it produced a new type of adhesive trap. So down here, you can actually see this sort of little hourglass-shaped knob, and then around it is a droplet of adhesive. This stuff is incredibly potent. So any nematode that contacts this is instantly glued down. What you actually see here in this photo is a little gruesome, but something that Drechsler mentioned specifically in his work. Often nematodes will thrash and try to escape so hard after touching these that they will sort of pull out of their own skin. So what you see here is basically the skin of the nematode adhered down by these two traps, and then it's actually sort of degloved itself trying to escape. And ultimately it won't be able to get away. Those pegs will penetrate and eat it from the inside out. Sometimes they're also poisonous, but they were primarily just adhesive. And remarkably, it also allowed them to capture things like amoeba, rotifers, tardigrades. It opened up for them a bunch of different species where the nooses really only target nematodes. After Dreschler discovered Nematoctonus, gave it a genus name, because it was so efficient at killing a wide range of nematodes and a number of life stages. It wasn't just capturing young nematodes, it was capturing and killing all the nematodes. He started using it in the context of agriculture to deal with things like gall-forming nematodes that would attack crops. Other researchers started specifically studying the genus Nematoctonus. These are just some quick examples of the glue traps functioning for amoeba over here. And then we got rotifers all being attacked here, very dead digested amoeba here. But Nematoctonus was remarkable. This actually turned out to be a very large genus. People were describing a lot of new species from it. Some of them would have clamps all over their mycelium, so what appeared to be a sexual fungus. However, they seemed to only be producing asexual spores. This is a great example of adhesive asexual spores produced by Nematoctonus. Sometimes a nematode would just touch these adhesive spores, which might be littered everywhere, not even attached to mycelium. The spores would stick, especially concentrated around the mouth, because that's where nematodes will sort of probe. And then the nematode can't do anything about it. Even if it can still move, still eat, etc., it's really sort of a time bomb because it will grow down into the mouth and consume it to form a new colony. They saw sort of this range of adaptation in Nematoctonus where you'd have some that barely produced any mycelium. They would pretty much spread as spores, consume a nematode, and then just coat the outside of its body in more spores. There wasn't even extensive mycelium outside of the nematode. It was almost more like a bacterial infection or something, like so reduced, so highly adapted to consuming nematodes that it didn't have a normal life cycle at all. It didn't seem to need sexual reproduction. Whereas we could go all the way to the other end and find these on wood, consuming wood as well as nematodes, and having clamps, extensive large mycelium, and you get this really interesting like range of adaptation for consuming nematodes. Like the strategies here were intense. To sort of tie this all together, something really interesting happened. Barron was one of these mycologists working on Nematoctonus in the agricultural context. He was sent a sample of Nematoctonus from some farmyard, some like farmland soil. He cultured it and it actually fruited on the plate. Now this is important because up until then, we'd been considering these asexual, despite the fact that they formed clamps on their mycelium, which is a sexual structure for basidiomycetes. We just assumed they were reduced, that they only did the asexual thing. It fruited on agar, one of his little petri dishes. He couldn't identify it. So we sort of sent it around, crowdsourced with other contemporary mycologists of the time, and they actually came back and agreed that it was Hohenboehlia. So the little fruit body that had formed on this Nematoctonus culture was in fact Hohenboehlia. Probably the most common species we're familiar with is Hohenboehlia petaloides. I've heard names like the flower pot oysterling. It's really common in like mulchy soil or wood chips or flower pots indoors, potted plants. But this was huge. There's something called an anamorph and a teleomorph sort of system in describing fungi. An anamorph is an asexual fungus. So one that doesn't ever form sexual fruit bodies, just does asexual spores. And a teleomorph is a sexual fungus. So like an oyster mushroom or a bolete, etc., that's forming Basidiospores would be a teleomorph. Now, this was really interesting because researchers had been looking at Nematoctonus on its own for decades at this point, decades and decades and decades. Other researchers had been describing huge amounts of new Hohenboehelia species from the tropics, from the US, from all over. So we had researchers looking at this from either end. And once Barron finally made this connection, we started culturing Hohenboehelia we found, and every single Hohenboehelia that's cultured produces a Nematoctonus. So all the Hohenboehelia that had been going by their own names, all the Nematoctonus that had been going by their own names, they slowly started getting linked together because we could culture a Hohenboehelia, record the morphology of the Nematoctonus it produced, and match it up to the Nematoctonus that had been produced or described on its own separately. So we were connecting the anamorph and the teleomorph. And so far, every single Hohenboehelia that's been cultured has produced a Nematoctonus. There's still a bunch of Nematoctonus out there that we haven't ever been able to get fruiting that still go under the name Nematoctonus, despite the fact we know it's actually a Hohenboehelia. There's a one name, one fungus ruling in fungal taxonomy and nomenclature where we can't actually keep calling things two different names. So technically, Nematoctonus doesn't exist anymore. They're all Hohenboehelia. We're just still matching them up over time. So it was really, really interesting to see this. Now, sort of where this ties into like modern families as well. At the time, Tricholomataceae was a messy family. Tricholomataceae had a whole bunch of genera thrown into it that didn't make much sense. Pretty much anything that was what you'd call pleurotoid, so that sort of lateral shape, decurrent gills, things like Philloporus, Pleurotus, Hohenboehelia, a bunch were just lumped in there. They were lighter spored, and we knew it was a bad family. We knew it wasn't what you call monophilous. We knew there was species in there or genera in there that needed to be separated into their own groups. A researcher called Thorne pointed out that at the time, there was only 2 groups of Basidiomycetes they knew of that were predatory. We know more now, but at the time, there was only 2. Pleurotus producing the toxocysts and Hohenboehelia producing the adhesive traps. He proposed a really, really interesting sort of argument that Pleurotus and Hohenboehelia belonged in the same family, not only because of this trait, but because of morphology, genetics, etc. And his argument ended up being supported. They were able to get them lumped into the same family. What was unique about this is that it wasn't just morphology and genetics that put them together. This was pretty much the only case where behavior, predatory behavior, had been used to put species together, which I thought was amazing. They even suggested there might be a common ancestor linking Pleurotus and Hohenboehelia as Pleurotaceae, which is a family that's still here today with these 2 genera in it. Now, years later, much closer to modern times, we found Hohenboehelia canadensis, which on its mycelium produced sticky traps and Pleurotus toxocysts. It had both types of traps on its mycelium, and they proposed that might have been the link. That might have been an example of what this family looked like before it started diverging, where you have Hohenboehelia, some of which are so specialized in nematodes, they don't really produce large mycelia. And then for Pleurotus, you have ones that are basically prioritized consuming wood. Their finding of this species really supports that predation sort of formed this family grouping, that both groups radiated out from a strategy of predation, and genetics for phylogeny placed it right down at the base of Hohenboehelia, right next to the divergence. So like, it was pretty wild to see Nematoctonus from Dreschler to Hohenboehelia by Barron, and then thorn going from all that to our modern family of Pleurotaceae. This research is still very ongoing. Unfortunately, a lot of this study took place at sort of a time where we actually funded exploration for the sake of exploration. We encouraged people to look and do new things. And a lot of this was just bare bones microscopy on agar plates to see these things. That's something we don't really do anymore. We don't really plate a well-known species on water agar and expose it to nematodes to see what happens. We don't really put random litter or soil samples on water agar to see what happens. Like, this sort of exploratory stuff has really fallen off. We haven't really had new predatory species described for a while, despite the fact that this is an ancient strategy and almost definitely all over the kingdom. There's a lot more that I think we could probably find if we keep looking. And that's why I'm so inspired by Dreschler's method that was so simple, using just water agar and microscopy to like observe these interactions. You can literally watch nematodes getting attacked and trapped by fungi. You can watch amoebas try to eat one of these sticky traps. And despite the fact that amoebas are blobs, you think they'd be able to get away, get stuck to the mycelium, trapped and melted from the inside out, where amoebas are usually the ones doing the melting. So like, I really encourage people to sort of take away the idea that a lot of this can be explored. There's still very much a lot to be found for things like this. Things like the spiky balls with Caprinus, we only found by harassing well-known species, and nobody's exactly doing that in academic mycology. But this is absolutely something you can do at home, sort of in the context of a hobbyist mycologist. There has been a lot of success using predatory species to deal with plant parasitic nematodes, human parasitic nematodes, animal parasitic nematodes. There's some really, really interesting applications with Nematoctonus, now Hohenboehlia. And Hohenboehlia in particular are so aggressive that if you were to slurry or grow Petaloides, Hohenboehlia petaloides on purpose, you could absolutely use it to treat nematode problems you were having in your garden. I don't really like the common name flowerpot dapperling, so I've just been calling Petaloides the nematode eater for a while now. I think it makes more sense with its ecology, and it's just kind of cooler. But that really is sort of some of the crazy strategies that are out there, how this affected modern taxonomy, and just, I love this sort of like this isn't a story that was ever laid out in one place. This is like, I originally sort of stumbled upon this doing reading, and you find Zopf, then you find Dreschler's papers, then you find Barrett and Thorne's papers, and all of a sudden you realize like, oh, all these people have been like talking to each other, and there's like a contemporary concept of what we're all looking at, what's going on, and you can see like this really cool thing sort of grow out of the mycological community at the time. I think I'm probably ready for questions on that. I don't want to go
That'd be great.
too long on that. Let's see,
So
how
this
long
is
did
about, this is good timing. This is good
Okay.
timing, right?
Perfect, yeah. So yeah, questions. Do one of you, I can read them out of chat, or
Well, I can tell you.
Okay.
So someone was asking about the makeup of the toxin in the toxocysts, and I actually found an article that is publicly available
Yeah,
that says the compound that's in there. So I posted
Yeah.
that in the chat.
awesome. Yeah, there's a few
And-
out there. I think one of the big ones is called pleurotin. But there's a bunch that are used very effectively against nematodes.
Right.
I know some of them have been looked at in the context of like possibly being medications for humans as well.
Cool. So have you done any of this, like trying yourself, any experiments yourself, growing stuff on water agar and just seeing what you get?
Yeah, I really used a lot of water agar when I was at LCC. partially because I was using it to clone fungi, but conveniently, when I was doing wild clones of a lot of these things, you have mycelium growing out on water agar, and there's a ton of different species that produce structures we don't know what they do. You have to do, there's sort of this, when you want to confirm nematocidal tendencies, you have to do assays involving known numbers of larvae introduced to a plate, and then you basically count clearance, like what percent of the population dies. When it comes down to mechanism, so like the stephanocysts or the snares, you are watching nematodes be trapped and killed. But for the most part, I've just cultured Nematoctonus from the Hohenboehlia to look at, because we don't even have names for a lot of the anamorphs of the species. At this point, because we know they're the same, we don't exactly like still have to describe them. But because every single Hohenboehlia you grow produces a unique structure and mycelium for killing nematodes, I still think it's worth exploring. So it's probably the easiest one you can get a hold of. That is easy to clone, and then the Arthrobotrys are super widespread. If you can often catch it just by doing the leaf litter method on water agar, but you can also deliberately spike plates with nematodes and sort of inoculate progressively until you get something that will antagonize them. And it's usually the Arthrobotrys that'll pop up.
So Kel is asking, what do they think the toxins might be used for in medicine?
It's sort of complex. So when you're talking about like pharmaceutical exploration, one of the first things you're just looking for is targeted activity. So even if something is extremely poisonous, like totally able to shut down voltage channels on neurons, for example, which is catastrophic, it paralyzes or kills things. When we see that compound is extremely selective, that is huge in the world of pharmaceuticals. Whether or not that individual compound can be used as is, often what we'll do is we'll research those, find out why they're selective, and be able to actually create derivatives or new versions or similar drugs that act on the same mechanism. So say you're concerned with something like Alzheimer's, you may be able to create links between the formation of certain plaques. I know that research has been kind of contentious lately, but you may be able to find relationships between certain receptors, certain channels, and formation of those plaques, and then go through your catalog of drugs available that are involved with those channels. And from there, sort of have a ground for moving into wider diversity of chemicals. I know some of these are weird fatty acids or sort of oily compounds, and I don't actually even know if we know how those ones work specifically, but the most well-studied ones are the neurotoxin, nematode-specific neurotoxins. And thankfully, nematodes are widely used as model organisms for animals in general, because they're sort of like the smallest, simplest thing we can have that shares most of the way our cells work. I hope that answers the question. Oh, Cornelia, I think you're muted.
Sam, Karen just posted a question about what role nematodes play in the soil. And then, because I know there are good nematodes actually, like they're not all bad guys.
It's complex. It's like not exactly something you could sum up. Like when I say that they're in basically every single ecological role, that's not an exaggeration. Like every nematode tends to have a different strategy. There's things like vinegar eels, tiny little nematodes that grow in fermenting fruit or in human fermentations in vinegar. All they do is consume acetobacteria. They're just a member of that community acting as a predator. You have other ones which effectively destroy plant roots. They'll either directly eat plant roots, sometimes they'll spread viruses or bacteria to plant roots, or some of them form galls where they'll sort of burrow into a plant root and or even further up into the stem and leaves sometimes, but they'll form these burrows or galls in the plant tissue, and the hormonal changes can disrupt the entire plant or kill it. Especially some of the bacteria vectored around by nematodes are devastating for human crops. But then there's others which are literally just detritivores. All they do is eat anything and break it down and further sort of the carbon chain and breakdown in the ecosystem. There's others that only just suck the contents out of fungal cells. Others prey on other nematodes. It's like, it's all over the place. Some won't really ever be found out of animals, whether it's GI tract or eyes or skin, acariasis, or no, wait, yeah, I think that's the name. Ascariasis, that's it. Really, you can't sum them up exactly as good or bad. Humans, probably most of our interactions with nematodes have been negative, but soil certainly wouldn't work without them.
And then another question is about whether you know which Pleurotus cultivars are the most aggressive nematode eaters.
I've never really seen enough papers with the same metric to put it into a good context for comparison, but probably most of the literature out there is on Pleurotus pulmonarius. That's one with confirmed strong nematode eating habits. In cultivation, it's probably not a behavior you're necessarily going to see unless it's nutrient deprived. So like on water agar, or maybe not in the absence of nematodes. Sometimes these predatory fungi don't want to waste resources unless they can smell or sense that their prey is there. They'll rely on that detection before they ever invest in the structures to trap them.
Okay, we probably should move on to fluorescent fungi at this point. And
Yeah.
I guess if anybody has any more questions about this, you could go ahead and post them in the chat and we can come back, circle around. So thank you. That was fascinating.
Yeah, absolutely. I really enjoy talking about it. Is it okay if I step away for like 2 minutes and come right back?
Oh, sure.
Yeah, all right. I'll be right back.
Oh, so Karen Hurtubise, who works at the John C. Campbell Folk School, she posted something about growing a specific variety of marigolds to deter nematodes. But, you know, as far as compared to this discussion, she said it seems oversimplified. So Kathy, you're asking about whether we could use Pleurotus as food and nematicide control. And I think Trad has talked about that possibly in his book a little bit too. So.
And now is a good time for a bathroom break for anyone who
Right.
has been sitting with their legs crossed.
Yeah, if you can get online, there are actually YouTube videos. And I remember the first time I watched one of those, you know, where Sidnee is talking about those adhesive points. I mean, it was like you were looking at a human thrashing around, and the only point of contact was like the very tip of the pinky, and it couldn't get away. Like it was just so intense looking to watch this thing. So I hadn't heard about the degloving part, and it makes sense if it's that sticky that.
It's absolutely brutal. And definitely like seeing the giant ones captured, like, you know, a nematode this size relative to the mycelium being taken down by these tiny little hyphae, it conveys exactly how potent some of these toxins are. And then ones like this, that is, I mean, if you put it in the perspective of like an animal with this mass relative to what it was hunting, it just doesn't exist. Like these fungi are so well adapted, they're taking down things the size of whales.
Right.
But
so,
yeah. Unless there were any more super pressing questions, I think I can probably get on to the fluorescent fungi. This one usually moves pretty quick, so hopefully I don't go too far over. So, fluorescent fungi. A lot of us are probably familiar with the concept of glowing mushrooms, have seen photos of glowing fungi in one context or another. I'm primarily going to be talking about the most common way that mushrooms can glow, and that is fluorescence. So just a general concept of what fluorescence is. It's all over the place. Fluorescence is an extremely widespread phenomenon, something in nature and human life that we can see all over the place. It's used in science extensively, things like fluorescence microscopy and tagging, hydrology, studying the flow of water. We may stain water with fluorescent dyes. This is fluorescein, a super potent fluorescent pigment. It's used in art. I don't really see these fluorescent posters as often anymore, which is kind of sad. And everywhere in the natural world, not only organisms like this caterpillar here, but minerals as well, naturally occurring chemicals. These are euperlites, a really, really well-known type of fluorescent mineral. I get my states mixed up all the time. I believe that's Michigan, all around the lakes there. All fluorescence basically works on the same mechanism. I won't get too far into this, but the general concept is an exchange of energy. So you'll have UV excitation, so ultraviolet light that excites a molecule. It could be a pigment, an organism, a mineral, just a molecule. Will absorb ultraviolet light and go into what's called an excited state. It'll have all this energy that it doesn't know what to do with. It's basically sort of resonating. That will get re-released. The way it gets rid of that energy is as light and heat. So you're always losing some of that energy to vibration, heat. The light color that you get spit back out will always be a higher wavelength, a lower energy wavelength from what it was excited with. So we'll excite it with UV, and then it'll spit out anywhere in the rainbow. Talking about the electromagnetic spectrum, all of the longer colors could potentially show up with fluorescence. It occurs in every single color of the rainbow. Some fluorescence is actually just infrared too, not even something we can see with our eyes. But the key is that absorption of energy from a high energy light source and then re-emission at any of the colors of our rainbow. Before we get too far, this is a super common mix-up. People often confuse things that glow in the dark because there's different mechanisms. There's really three big ones. There's fluorescence, there's bioluminescence. Those are really the only two that matter in the context of glowing organisms. And then for things like minerals and man-made substances, there's also phosphorescence. Now, fluorescence cannot be seen without an excitation light. So you can't really visualize fluorescence without UV or deep blue light from somewhere. These things will just look dark in the dark. They won't glow on their own. Bioluminescence is a biological process, strictly, and it does not require input light. So bioluminescence actually uses energy produced by the metabolism of the organism to produce light at a very specific color. So what we have here is a bioluminescent mushroom. This is cultivated Pinellus stypticus on the left, and on the right, this is Albo gymnopilus, fluorescing blue. With fungi, if it's glowing on its own, it's bioluminescent, always. If it only glows when you have a UV light, that's fluorescence. Now, fungi basically all use the same enzymatic system to produce light. So far as we've found all fungi produce the exact same color of bioluminescent light, which is this sort of ghostly green on the left, whereas fluorescence can come in any color. Purple's, you don't really exactly see purple fluorescence, but any color of the rainbow, more or less. Going into UV light, just a brief explanation of like wavelengths. So this is basically the electromagnetic spectrum of what we consider light. Everything that we can see just with our eyes as they are is what we call visible light, from roughly 400 to 780 nanometers. Now that measurement at the bottom in nanometers refers to the length of the wave of the photon. You'll often see wavelength mentioned when it comes to light sources of UV. It's also sort of easier to just know the categories of UV that we're working with, because some of them are hazardous. UV is basically divided into 3 categories. These are all mostly outside of the visible range. We can see a little bit down to like 365, 360, but it starts getting really, really dim. And it's a light that we just can't pick up. Bees can see it, a number of bugs can see it, some crustaceans and animals, but humans can't really pick up on these lights, which can be dangerous in some cases. From about 315 to 400 nanometers, we have what's called UVA. This is the least dangerous type of ultraviolet light. And when you're talking about, when you hear the word black light, it's always going to be UVA. Black light in particular actually refers mostly to 395 nanometer light. So this is barely UV. It's pretty much just a deep, deep purple light. Going a little further, the other common light you'll see is 365 nanometers. This is because when it comes to little LED flashlights, there's really 2 primary chips on the market, one for 395 nanometers and one for 365 nanometers. This does matter if you're considering the purchase. 395 is cheap. It's probably the most commonly available. And if you just get yourself a UV light without knowing the wavelength, chances are it's just going to be a 395. It's definitely the most common. And it works for a lot of stuff. 365 is less visible to us, and it doesn't have that purple hue. So like black light, 395 is distinctly purple. And when you're looking at stuff with it, you'll often see the fluorescence tinged a bit purple. And if you have something that's only really weakly fluorescent, it might even be washed out completely and just look purple. When you go down in wavelength a little bit further outside the range of what we can see, you'll have 365 nanometers, which appears very dim and more bluish to our eyes. These lights are often very, very bright. They just don't look like it because we just can't see it. It's almost like we're blind to a spotlight with some of the nicer ones. Because they don't put out as much visible light, you'll be able to see true color a lot better with fluorescence. It's better for photography in general. And also because it's a lower wavelength and more energetic, it actually makes more things fluoresce too. There's a lot of things that will only fluoresce under 365 and not under 395. And then getting into UVB is where it kind of starts to get dangerous. UVA lights basically pose the same hazards as visible light. Any super bright light can damage your eyes with, it's a term called flash blindness. Your eyes are being damaged just by sheer amount of light hitting. It's not a specific radiation damage or mutagenic damage. It's just outright overstimulation and burnout. When you get into UVB, it starts to get more dangerous. UVB are the wavelengths in sunlight that cause sunburns, radiation damage. They can generate ozone to some degree. And you also can't see it. UVB is just invisible to us. So it's really easy to accidentally be shining it on yourself or someone else without realizing it. Thankfully, other than tanning beds, we don't really encounter UVB. Nobody really sells UVB flashlights. So the flashlights we work with, just don't shine it in your eyes or your friend's eyes or onto super reflective surfaces where it could get in your eyes, and you're basically fine. UVC is not really something, they're actually sold as germicidal lamps. It is so energetic, it can pretty much immediately cause burns if the light is bright enough. It causes cancer, immediate eye and skin damage, and it's energetic enough that it will actually form ozone in the air that it passes through. It will convert oxygen to ozone. And so even if you're not shining it directly on yourself, you can still get damaged just by being in the same unventilated room as your light source. Everything fluoresces under UVC, but it should never be handled without eye and skin protection in a ventilated outdoor setting. I don't really recommend anybody take a germicidal lamp out to the woods because they're quite dangerous. So really just 395 and 365 are pretty much all you have to remember. If it looks purple, it's probably a 395. If it looks blue, 365. And now going into the actual fungi, this is probably one of the more well-known fluorescent mushrooms. So this is Hyphiloma fasciculare. People call them the sulfur tuft, and even in normal daylight, they look neon. They look striking. It's not surprising at all that they would glow so brightly under UV. It's probably one of the main ones you'll notice if you're ever using a UV light out in the woods. It'll certainly be one of the brightest on the table if you're shining it at all your collections at once. And because this species is actually daylight fluorescent, so just the UV and sunlight is enough to make this glow, which is why it appears that sort of like extra bright neon when you see it in person. This is one of the few cases where I think you could potentially make the argument that it had functionality. Just the fluorescence itself might have functionality potentially for bringing in vectors to distribute spores, attracting insects. Very few fluorescent mushrooms are daylight fluorescent. This is probably one of the only ones where I think we might be able to make the argument that fluorescence is actually their goal. But this is always a showstopper when you see it out in the woods at night with a UV light. And I don't actually know the name for the pigment in that one that makes it so bright. This is a great example of sort of the opposite. So this is Cortinarius clandestinus. This is a pretty drab, boring looking Cortinarius. Over on the right, where'd my mouse go? Over on the right is the mushroom in daylight, just sort of drab, browny brown. Over on the left, when you hit it with a 365, intense, like stoplight yellow fluorescence. And this photo is in daylight, and it is still this bright. This mushroom is absolutely comparable to Hyphiloma fasciculare when you're lighting it up at night. This is actually due to the presence of a chemical called leprosybin, and conveniently, the whole section, so Cortinarius is sort of divided into sections, may or may not be risen to genus level, but there's a section, Leprosybie, and every member of Leprosybie contains the chemical leprosybin. Every member of Leprosybie glows this exact same color under UV. This can actually be a diagnostic tool for getting to a section within a really difficult group of species. And there's another group, the Dermosybie, the Dyer's quartz, that also show fluorescence, but they're not the same shade. There's a super variable, it's all over the place. They do not contain this specific pigment, even though they do glow a bunch of pretty reds and oranges and pinks. This is another great example of sort of unexpected fluorescence. So these are actually hydnum. I'm not sure if they're common in Georgia or not, but this is one of our most popular edibles here in Oregon, hedgehogs. And despite the fact that they're bright orangey yellow in normal light, they show distinct pink purple fluorescence. In particular, anywhere they're damaged or bruised, they'll show this color. Sometimes randomly, I'll see this on chanterelles too. I have no idea why it doesn't seem to be a consistent reaction. This is a great example of one of the yellow boletes. Most of those neon yellow bright species will glow. There's a range of compounds responsible for this color in particular, but interestingly, you can actually dye yarn fluorescent with these. They'll be yellow in normal light and then fluorescent yellow under UV light. When it comes to uses for these pigments, that's actually sort of one of the more popular, newer uses is fluorescent dyeing with mushrooms. One of the most iconic species in dyeing in general, Phaeolus schweinitzii, the dyer's polypore, produces incredibly potent fluorescence on any yarn that's dyed with it. Same thing with Cortinarius, the dermosity. But it sort of gets into the application side of fluorescence. Taxonomy-wise, we're kind of behind when it comes to mushrooms. So lichenologists have been observing and recording and characterizing fluorescence in lichens for a long time, basically since the 50s. In particular, they understood, because chemical differences in lichens are so important in lichen ID, what they call chemotaxonomy, they noticed fluorescence reactions would vary based on the pigments that were present, or based on the fluorescent color that was present. So they'd be able to essentially see a particular color of fluorescence, know that a chemical was there, and use that to ID it, to even erect large taxonomic changes. Since the 50s, they've been documenting these reactions. We don't really have this for mushrooms, despite the fact that fluorescence reactions are common and distinct in many cases. We haven't systematized it very well yet. Pretty much the only group that has sort of formal recorded fluorescence reactions where it actually can show up in a key is Cortinarius in dermosity and leproside. The fluorescence can be used that way. It's basically the only example out of all the mushroom species we have. A big part of that is that we're missing the observation part. We just haven't looked at most mushrooms under UV ever. And so we don't have the information to create any systematic approach to this. We're just lacking the info. You know, lichenologists are dealing with species that are often so cryptic and difficult to tell apart. They go down to the metabolism to tell them apart and what's going on in chemotaxonomy. And we haven't made that leap for most mushrooms yet. One group where this will probably be happening in the next few years, Matt Schenk has actually been working on characterizing and documenting the fluorescence reactions in this group. You'd probably never guess what these were, just seeing their fluorescence. These are actually Ganoderma in the group of what people would call American reishi, the lacquer top conchs. They show brilliant fluorescence in a huge range of colors. He has been basically accumulating photos of different species, ideally with sequence data to back them up, because we have a nightmare mess of species complexes in Ganoderma in the US, and he's convinced fluorescence might help us key them out a lot better. He's at the stage where he's basically just accumulating observations where a species will be fully identified, the fluorescence reaction it has will be documented, and you can start making those connections. It's not until you have that information on traits that you can actually start to create keys that might use this. But that's Matt Schenk, sort of a Ganoderma nerd. This is an interesting one. So this is actually Boletus edulis. I think it might have been Grand edulis. But Boletus edulis, basically, down here in normal light on the bottom, you see pretty much just standard bolete coloring. Under 365, they'll often have areas of their tissue that are insanely bright, this sort of weird orangey, almost greenish tinged fluorescence. the color is identical in all the species that I've seen this in. Caloboletus, Boletus, Xerocamelus, a lot of different groups. You'll see this sort of same weird shade of orange, almost definitely due to the presence of one shared chemical. Now, what I thought was particularly interesting about this reaction that hasn't really been characterized is sort of the areas you'll see it in. In Boletus edulis, you'll often see it in the pores, maybe in the stipe base. In other species, it will only be the stipe base or sometimes the surface of the cap. It's sort of all over the place. And what that's reflecting is actual metabolic differences. They're choosing to produce whatever this compound is only in certain places for a specific function. I was absolutely tickled to find that Caloboletus marshii, a species that I find in Eugene under oaks, the mushroom itself doesn't really have this fluorescence anywhere. But if you dig underneath it to look at the mycelium has this same weird greenish orange fluorescence. These are rhizomorphs of Caloboletus, and they just stand right out. You can actually observe the fluorescent coating of mycelium on mycorrhizae. I had a lot of trouble photographing it, so these rhizomorphs, the huge ones, are kind of the best I could get for this. But another example of like this compound is in a totally different place for this species than like Boletus producing in the tissue. And knowing that these fluorescence reactions indicate presences of like the presence of a specific chemical, it is almost a little bit of a window into their metabolism, like where they're producing a chemical. And it brings up questions of what are they using it for? Why are they using it for one thing in their mycelium, one thing in their cap? Is this anti-predatory? We don't know. For the vast majority of fluorescence reactions, they haven't been explored. We don't even know the compound is responsible in a lot of cases, the pigment associated, let alone what that pigment is specifically being used for. This is a just sort of great example of that orange fluorescence taken to an insane degree. This is actually Pseudoboletus parasiticus, a parasitic bolete that attacks scleroderma. So over here, this is our parasitized scleroderma looking very sad. You can see it's being eaten on the inside. And then this is the bolete growing out of it. You might think you're looking at just an orange bolete, but this picture is all UV. All the color here is a UV fluorescence reaction. It does not look like this in visible light. Just a crazy concentration of pigment throughout these things. And the boletes are a great example of that sort of like hidden pigment concept or hidden fluorescence where, you know, Hyphiloma fasciculare, it's obvious that thing's going to glow under UV. But Boletus edulis, orange is not what you would expect. And so I sort of want to go into how you find these, how you can start gathering that data to characterize mushrooms so we can start learning more about fluorescence in actual mushrooms, not just lichens. It sort of boils down into what I've been calling night forays or night walks. I'm extremely excited that it sounds like the club is getting these together. Absolutely love that concept. Again, I think it's probably the first club I've heard of doing this in an organized fashion. And it's pretty simple overall. It basically consists of going to the woods in the dark and looking for fluorescent or possibly bioluminescent organisms, ideally fungi, but you end up seeing a lot of really cool things out there. Going into the planning and safety aspects of it, you want to choose your location and scout it in the daytime. This sort of serves two roles. You want to get familiar with the area, become familiar with the hazards. It lets you ensure that the path is clear, open to walk. It gives you kind of a spatial sense of where you are, because you will not really be able to keep your surroundings coordinated in pitch black darkness. Much easier to get lost. So this allows you to sort of select your location, avoid hazards like cliffs and rivers and eye level sharp branches. That's a big one. And then the other factor is confirming the presence of fungi. It's nice to be able to go out, see that there's things fruiting, and know that you're actually going to see some things out and about. You know, fungi and lichen aren't necessarily the only thing you'll find out there. Lichens are nice because you can see them any time of year, and they have some brilliant fluorescence reactions, but bugs, minerals, animals, possums fluoresce pink, all sorts of different things you want to kind of look out for so you can get some really interesting colors and reactions. After you've selected your location, I recommend not doing this alone unless it's like right down the street from your house, because it is incredibly easy to get lost in the dark. Bring a flashlight, a normal flashlight, as well as your UV lights. And I highly recommend either bringing a backup normal flashlight or spare batteries. You don't want to hike yourself out a half mile on a trail, have your lights fail or break or run out of battery, and be stranded in pitch black darkness trying to find your way back. Also, another reason where the group sort of comes in handy. Less likely to get lost, less likely to be stranded without light, and people actually know where you are and what you're doing. Wandering out into the woods alone at night can be a little dangerous, so groups are best for sure, or at least just a friend. When it comes to light selection for UV, I would recommend 365, but because some things fluoresce better under one or the other, I actually carry both. There's some lower tier, more affordable ones available online for like roughly 35, 30. That'll get you as far as you need. There's higher end ones closer to 100. Kind of a good range out there, but thankfully LEDs have come a long enough way where these are actually affordable and portable. If you use Amazon, I'd recommend looking around there. Tattoo and Convoy are both great brands. But if you were only to buy one light and you had to choose, I'd go with a 365. Just, it'll give you the best range of colors. Might be a tiny bit more pricey. If you're going out there to see bioluminescent fungi as well as fluorescent ones, because fluorescence is observed with a light, you don't need actual pitch black darkness. You can even do it twilight sometimes. You're actually illuminating things, and the light you get back is proportional to what you're putting in. Now, bioluminescence, there's a few species that are like glow stick bright, incredibly visible bright, but the vast majority of bioluminescent fungi are pretty dim. If you want to be able to see that, you need actual pitch black darkness, which is surprisingly hard to achieve. You're going to want a night with a new moon, so either waxing or waning, just tiny moon. If you can actually plan your time before or after moonset, moonrise, where the moon's not even in the sky, that's ideal, because just moonlight can interfere with this. Once your eyes actually fully adjust, you'll realize how bright everything is. You'll want to choose somewhere far away from any light pollution. I mean, you have to be more than a few miles away from a town before you stop noticing the like orange glow coming from the sky. If you can, kind of middle overcast, and then ideally you actually want a dense canopy above you to block out even more of the light from the sky. And then once you've achieved that pitch black darkness, situate yourself and just turn all lights off. You want zero illumination from any source, and just sit there for like 10 minutes. I've been stunned doing this. The amount of bioluminescent fungi and just organisms in general, bacteria and animals, has blown me away. I think the problem is that just not enough people go out into the woods in pitch black darkness and sit there for 10 minutes. But like the amount of organisms that have dim bioluminescence is insane. Just digging through soil, digging through wood, looking on the bark of trees, you'll start to notice glimmers everywhere. But that does take a little bit more planning just so you can ensure pitch black conditions. or one thing I'll do sometimes is just harvest things, whether it's a mycelium or the fruit body, take it home and then sit with it in pitch black darkness in like a bathroom or something so I can see it. Overall, fluorescence is a bit more accessible, easier to do spontaneously. As for citizen science and contribution, if you start getting into this, start photographing new species and specimens and their reactions. There's actually a list on Mushroom Observer. And then I believe there's also a project on INAP where you can attach photos to characterize the fluorescence of different species. But otherwise, just photograph these reactions when you see them. That alone, so long as you're able to share and relay that information on reaction, you're already helping contribute. Because we just haven't looked at most species. Even now that we are starting to get more popular night walks, more people interested in UV and fluorescence, we've still examined just the tiniest little sliver of what's out there. I think that probably covers everything I wanted to do about fluorescent fungi. I'd be happy to go through some of the questions in chat if we had any. Time-wise, yeah, perfect. 40 minutes. Name of the citizen scientist site, Mushroom Observer. You'll have to make an account, but then you can also upload them to iNaturalist as well. I believe there's projects, multiple projects you could add it to direct there, but just uploading those photos to iNat. So like normal light photos and just normal light photos and UV photos alongside, that'll already add information. Because when people go and look at your observations on iNat, they'll be able to see right then and there what the associated fluorescence reaction is.
So one of the things in conversation with you is you were saying, especially like for us going to be doing a night fluorescent walk, is that it's really good if you can walk the trail while it's still light first, so that you get a real, heading of what it's going to be like in that.
Yeah, I always recommend scouting the area in daylight. Like one of the best situations is like where I'll often do it is camping, because then I'll be able to like potentially hike out at twilight so I can see what's going on. By the time you're heading back, it's dark, and so you get to sort of like rewind the path in from a whole different viewpoint. Or just camping gets you out there at night and you can see it all. But I'd always check an area, just make sure you can actually see it in the day. Yeah, I'm really glad you're doing that. I think it sounds like an awesome opportunity.
And I'm trying to remember from your night walks at the festival, do you walk with regular like headlamp flashlight and then turn them off and do the UV, or how?
So typically what I'll do is I mean, it's a little nerdy, I guess, but I'll have one in each hand. So I'll have a normal flashlight in one hand and a UV light in the other. I'll sort of alternate them. So I'll use a normal light to navigate, to walk myself to like a bit of a new area, turn off the light. And then I have a Convoy C8. It's a pretty powerful sort of 365 spotlight. And I'll turn off my normal light and then scan the area with the 365 so that anything that glows in the area will stand right out. And then I'll just sort of be like slowly pacing my way through an area, making sure that I get to see everything in visible and UV light.
And so there's no issue of like eye adjustment from
no, not
regular
with
light?
fluorescence.
Okay.
With fluorescence, your eyes don't really need to be adjusted. Bioluminescence, absolutely. You have to choose a good area. And if you want to just observe where you are standing, you can just turn off all your lights and sit there for 10 minutes. You absolutely cannot turn them back on. You'll immediately ruin your night vision and you'll have to wait a whole other 10 minutes. What I've done is like I'll just turn off my lights and sort of crouch and then kind of like hands and knees around to dig through duff and logs and litter. And I found lots of stuff that way. So once your eyes are fully adjusted and you have pitch black darkness for bioluminescence, you really don't want to turn your lights on if you don't absolutely have to. It's definitely more difficult to do with larger groups of people. I can say that, but fluorescence is no problem because you have a UV light. It's very bright. You won't have to worry about adjustment.
So we did have someone ask, because we talked about the henna phalis. If you could talk about that process just a little bit before we move on to the dyeing video.
Yeah, that ties together. So like I mentioned, sort of how you can get a fluorescent dye with that bolete. One of the species where I've really worked with it, I have an interest in mushroom dyeing. I've been working now for a while on new dyeing methods, new ways to use dye fungi, new colors. And one thing I stumbled across was a new way to use the fluorescent property of the dyer's polypore, Phaeolus schweinitziae. This is a mushroom that contains hispidin and a few other fluorescent pigments. What I noticed while dyeing normally with it is it dramatically stained my hands and nails bright yellow green, but only under UV light. My hands weren't yellow tinged at all. In visible light, it was effectively almost an invisible ink for skin. After a bunch of playing around with that, safety research and background and just exploring, I finally sort of developed what I've been calling mushroom henna or Phaeolus henna. It's basically a paste that you apply to the skin in the same way you would standard plant-based henna, except this is all mushroom. It is only made from Phaeolus schweinitziae. You apply it to the skin, give it maybe 5 minutes, you wash it off, and you're left with nothing. It's just completely invisible under normal light. But when you get it under UV, you have neon glowing designs sort of anywhere you put it. So I've done a few workshops now, doing this with larger groups of people. I haven't started selling it yet. I probably will at some point, but I like the research more. But like at the International Fungi and Fiber Symposium, I did a workshop with this Phaeolus henna, and it's all based on the affinity of this fluorescent pigment for skin. So it is very, very potent at binding keratin, and totally invisible in normal light. So even if you're not very good at henna, the stakes are low. It's always been a lot of fun to play around with groups.
So I want to take just one moment to share my screen. So can you see the two different pictures?
I can see one photo right now.
Okay, so that's just our hands. Okay. And then this is our same hands a few minutes later.
That's a good photo.
That's what we did. That's what we did for Thanksgiving.
Love it. That was stuff you took back from the IFFS?
Yes, that's using those 2 plastic syringes that you gave us.
Awesome. Yeah, I love it. Here, I'll just start reading out of the comments. So someone asked how long it lasts. I've noticed it varies. I think it has to do with the rate of exfoliation of skin. It doesn't go quite as deep as standard henna. In my experience, it lasts anywhere from a day to a week. Usually it starts to really fade around day 2, but it also depends on location. It won't last as long on your hands as it would like up here on the soft part of your arm. It's sort of variable depending on where you put it, what your skin type is. And then, I mean, I would say I've never really had it go away in under a day, unless somebody's like purposely exfoliating with like a stone. I've tested that before. And yeah, that's Phaeolus schweinitzii, the dyer's polypore.
And in our conversation with you, said you were talking about it's better to use fresher, that
Yeah.
not use like dried powder.
Well, no, yeah, you can use dried powder, but what you do want is young specimens. So
Okay,
you
so
want-
you can dry a young specimen.
yeah, yeah. But the main thing is you want something that still has ideally that yellow-green band on the edge and still bright yellow-green pores. If it's at an age where that band is gone and the underside is really starting to go more brown than yellow, your yield it won't be anywhere near as bright. The pigment starts to polymerize into a brown polyphenol, and it's lost at that point. So the highest yield is young specimens.
Okay. So we're gonna need to wait till the spring to make some fresh.
I mean, if you have dried young Phaeolus, you can use it at any time.
I think most of our dried are old ones, so,
We have a few young ones.
okay.
We'll have to-
yeah, I mean, if it's dry and it still has yellow on it, it'll probably work. And even the old ones can work. It just won't be as vibrant. But
So
yeah.
that
So
could
we
be a
have-
project for the club.
We also had
Yeah.
a question about whether you worked with Pinellus stypticus, which is bioluminescent, which is different.
The only bioluminescent mushroom I've really worked with is Armillaria honey mushrooms. So while I was at LCC cloning, just like everything I came across, I cloned, I think, Solidipes, Melia, and Nabsnona, and oh, actually no, I think also Gallica and Ostoye. And a bunch of those glow very, very bright on agar. You'll have sort of like it glows brightest at the edge where it's growing or anywhere it's been damaged. So you'll be able to see sort of this green glowing ring. At certain stages in development, Armillaria can outshine Stypticus. The only real advantage to Stypticus is it's very steady and the whole thing, including the fruit body, glows. Armillaria, only the mycelium glows. But they're easy to clone and they're really cool looking because they also produce those like shoelace rhizomorphs.
And someone who was asking if you said possums glow fluorescent pink.
Yes, possums glow fluorescent pink. A lot of
possum?
animals, yeah, the fur, yeah. Their skin is more blue. And beaver teeth, beaver feet and hands, nutria teeth, nutria feet and hands, and organ flying squirrels, also bright hot pink.
So it'd be
A
interesting
lot of animals.
to see if there's a microbiome associated with these different colors or something like.
So there was actually a paper published on the flying squirrel. I'm not exactly a mammal person. I don't know if they actually identified what was going on, but there was a paper on the flying squirrel fluorescence. And that's anytime like we have the organ flying squirrels, and anytime I'm on a night walk, I always make sure to like stop and shine up into the trees just in case I come across one. It'd make my day for sure. But the possums are everywhere here. Those are easy to see. And so far they've all been pink. But we haven't checked most mammals. I've checked snakes and toads that fluoresce. A lot of bugs fluoresce. I don't know that I've ever checked any birds, but a lot of different things glow. That's the other thing is, I like to do night walks when there's mushrooms out. And I've even found like fluorescent slime molds too. But even when there's no mushrooms growing, no active fungi, you'll still see a lot of fluorescent things, fluorescent plants, bugs, animals, minerals, all kinds of cool stuff. I went to at Camp Westland, we were on the beach, and in the night we were fluorescent tide pooling, which also highly recommend if you can do it safely. Checked the cliffside, and the whole thing had veins of orange fluorescent quartz running through it. Absolutely stunning. It wasn't fungal, but it was probably the find of the night. Lots of cool things to see on these night hikes. Like I just encourage them in general because it's like a whole other aspect of nature that we don't really experience. Humans just don't go out to the woods at night very often anymore. We always have a light source.
Okay, I think it's time to move on to the 10 minutes or
Yeah,
so of the die. Yeah.
should be quick. I'll go ahead and
I will
share.
say Sidney has an excellent photo that he used in his talk of the class that he taught where people use
Oh
the
yeah, the
dye.
henna.
That's a
I could probably
beautiful
find that after I do this bit if you'd like.
Okay.
I think I have it saved somewhere. Let's see if this will work. Perfect. Oh my gosh, I nearly forgot. So in Savannah,
In
Georgia,
Savannah,
there
at the
is
home.
a UV art exhibition going on, just recently announced. Alan Rockefeller, Clay Emke, and Derek Larson are all working on this. It is at the Haunt, which I believe is like a restaurant.
Oh, Clay is the chef.
Yeah, it is. I like heard about this right before the talk, and so I just kind of threw this slide in there because I wanted to mention it. I've never seen any sort of fluorescence focused organism exhibit like this. So worth a mention because this sounds like it's going to be super cool. They just announced it. I'm not sure if the details are all out yet, but you should absolutely keep your eye on this. I believe that they have been posting about it on their Facebook page, The Haunt.
I think I know what we're doing for Christmas, dear.
Cool.
But yeah, quick mention on that because it's going to be amazing. Alan Rockefeller is probably the best fungal fluorescence photographer out there. And so this is just kind of a mention. Like I talked about, I've been interested in mushroom dyeing for a while now, and also just sort of in line with the henna, with a few different projects I've worked on. Like one of my focuses is finding new uses for fungi, just new methods, things that haven't really been done or looked at before. And so this was one case that I was just absolutely happy with. Like this, I'll just sort of briefly go over it, but it's really worth a mention. I think people will think it's cool. So just for some background, this is all based off of the bluing reaction of psychedelic psilocybe mushrooms. If you've ever seen these or read about them, you've probably heard about the blue staining reaction that a lot of psilocybe and other groups that also contain the alkaloids show, where when you damage the tissue or break them, or sometimes if they've seen like a frost, they'll go this deep blue or green color all over. The main two alkaloids that they contain are psilocybin and psilocin. Psilocin is actually the psychoactive one. Psilocybin acts as what's called a prodrug. The key difference is this little bit up here. It basically has a little extra piece, a phosphate group. It's what you would call phosphorylated, basically. And so this sits here, keeps it from being psychoactive, and then when you eat it, it'll dephosphorylate and become psychoactive. Now, we knew about the alkaloid chemistry, we've known about it for a long time. And we knew about the bluing reaction for a long time, but we did not know what the blue pigment was or how the blue pigment formed. We didn't know the reaction. We didn't know the purpose. It had been a mystery. Now, oh gosh, the years keep adding up. I think this was maybe 2 years ago, but relatively recently, a paper came out called Injury-Triggered Bluing Reactions in Psilocybe that actually found what the blue pigment was. It identified the blue color and then identified the mechanism that forms it. I'm not going to go too far into the chemistry because we don't have time. But basically, most of what's in the mushroom alkaloid-wise is psilocybin, the phosphorylated one. When you damage it, it rips off that phosphate group and you're just left with psilocin. So when you damage it, it all jumps into psilocin. And then a separate enzyme called a lacase, an oxidizing enzyme, oxidizes it. And once it's oxidized, psilocin molecules will actually link together. The first thing you'll get is a dimer or two linked together. and then more psilocin can come and just jump onto that until you get a bigger and bigger and bigger molecule, and it basically gets bluer and bluer and bluer, almost until it approaches this sort of blue-black color. But this was huge. We'd known about the bluing for a long time. We did not know that the blue pigment was literally formed from the psychoactive alkaloids. We have the structure and the mechanism. Now, when I read this paper, it got me thinking about the oxidation system used for a popular pigment, indigo. Indigo also works on some of the same principles of reduction and oxidation. And so I just started experimenting. They noted that this blue pigment was remarkably stable. It bound to protein as it formed. And so all of these pieces sort of started to fit together on how I might be able to get a pigment out of it. Now, I was in particular motivated because blue pigments are not very common in the mushroom dye world. There's really only one primary group of actual mushrooms that you can get blues from. That's things like hydnellum, sarcodon, etc. A lot of the tooth fungi. All of them are mycorrhizal, and all of them are not outrageously abundant. As mushroom dyeing has been getting more and more popular, there's been a lot of concerns about the availability of these blues relative to how much of the mushrooms are actually out there, because they are mycorrhizal. There's been some concerns over harvesting, but scarcity has been a problem with these, just with our current amount of dyers. As it gets more popular, we're really worried about blues not being much of an option. So this stood out to me as a new potential route for blue pigments. What I did is I basically replicated each step of this cycle, the dephosphorylation, the oxidation. You can do the oxidation enzymatically or inorganically with oxidizing agents. I've used other mushrooms to catalyze this reaction. Also, I can't get too far into it with our little time, but I wanted to show what this looks like. So basically, it's a two-part reaction that I do where I'll extract the alkaloids, turn it all into psilocin, because that's what actually is a precursor to the blue. And then I will have the psilocin mixture, and then basically the enzyme or catalyst solution. It's a two-part thing. In this case, I was actually using enzymes harvested from turkey tail mushrooms. They have an oxidative enzyme that manages to work very similar to the native enzyme that does this gluing reaction. So I immerse yarn in the psilocin mixture, just on its own. And then what you'll see is me taking the yarn out, adding the catalyst, putting it back, mixing a little bit of oxygen in, because this does require oxygen. And then the color change to blue is very dramatic, and this sticks right to the yarn. This is sped up. And that darkening happens right away. The longer you leave this, really the darker it gets. Because of the way this pigment adheres to yarn, you can actually do multiple layers similar to indigo. So your first pass, you end up with sort of a robin's egg, really light sky blue color, and you can actually repeat this process and get it darker and darker and darker. One more time, and then I'll actually stop screen sharing, and I have examples of the yarn right here I can show. But I figured, you know, we thought this would be worth a quick mention. Nobody's ever done this before. It's all been research that I have sort of been working on for a while. And I first announced it at the IFFS Fungi and Fiber Symposium. And I was really happy with the responses, because like, not only is it cool to use this group of mushrooms for dye where nobody had thought they could be used for dye, but also it offers a blue, a pretty scarce pigment in mushroom dyeing. So I can stop sharing, and then I don't know how well the lighting in here is going to show this at all. But so this one is halfway in a hat. but I have this blue right here, sort of like, you can see it, I guess, contrasted against the other colors. This is a half done hat. And then in this finished one, just a nice lighter blue band. I really like this color. It is a different shade than the one you get from Hidnellums, which tends to be a more greenish tinged. But this, in the proof of concept sense, for me, was just super exciting because, like, I came up with the theory after reading that paper, and then to see it actually like work out was amazing. Like going from that on paper to reality, that change was really, really cool to me. And I have been kind of like, I normally post like a lot of my experiments on Facebook regarding dye and just everything in general. But this has mostly been like sort of just notebooks. I am actually probably at some point in the next couple of months going to be releasing a zine covering this methodology, like the instructions, ratios, walkthrough, the mechanisms of how it works, where I'm at in the research, so I can sort of leave a starting point for anybody who else who wants to pick it up and do their own study on it. And at some point, it will probably also be published in Fungi magazine, but the zine is the next thing. And I'm probably going to be updating that on my Patreon, but mostly right now, just the zine. I think that basically covers the Psilocybe blues. Happy to take questions on it if we have time.
So have you, I mean, you know, you basically said that you're the first one to kind of, as much as you know, do this kind of experiment. Has it been out there where you've had scientists or citizen scientists like say, can we explore this more with you or whatever?
I haven't really shared it with anybody to that level. Part of it is that it's all still very experimental. Practically every time I rerun this, I find changes, ways to improve it. And so I've sort of been waiting until I have a nice condensed method that I feel comfortable giving people. Because at least certainly the first few times it was finicky enough that like somebody could probably easily try to do it and mess it up. It really is reminiscent of like indigo dyeing in a lot of ways, where if you're making your own plant indigo from scratch, people mess that up all the time. And so I'm really trying to get a nice foolproof method out there. As for the chemistry, I mean, it basically is all described in 2 papers. I am just adapting the mechanisms that they've laid out in order to create a pigment that can actually be adhered to things. It's sort of like, I think honestly, the main unique thing about it is just the new use. The fact that it does have a functionality in the dye world. And so things like the zine or what's his face? Oh my gosh, I can't remember his name. The man associated with Soma, is it Art Woodhouse? I think it's something like that. He was the one who has been talking to me about Fungi magazine, but for now, I really just want to get the method out there, have people experimenting with this on their own, then we can sort of all collect the information together. But I've really been prioritizing the method that uses 2 species, so turkey tail and psilocybe, to do it.
How do you get around the legality issues? Because psilocybe is illegal in Georgia.
Well, in Oregon, it's pretty thoroughly decriminalized. They don't really pursue anybody here who's not operating like a gigantic interstate trafficking ring. So it's thankfully extremely casual here. There's also the advantage that they just kind of grow wild everywhere in my town. For other states, unfortunately, I'm not sure there's much other than advocating. I think that in many states that are sort of on the border, projects like this, where technically the final product, the dyed fabric and the pigment itself, is non-psychoactive. It's not a scheduled chemical. The end product you're actually destroying the psychoactive component to produce it. So I think for areas where general culture, legality, etc., is sort of like on that line, this could represent something to almost casualize it, a new way to handle them that's not associated with, you know, drug use. A lot of people have negative connotations around what they envision drug use as and tend to smear that all over mushrooms. You know, they're demonizing mushrooms because they can sometimes demonize people that use them. And I think something like this is a perfect opportunity to show there's more to it. Their chemistry, their uses, reasons to have them. But it's something that doesn't take much and can be done at home in small batches quickly, easily.
I noticed you were not wearing gloves, and I have to wonder, does it get absorbed through your skin? Did you sleep really well that night?
So there is some level of dermal absorption, but this is a really dilute solution, relative. So I would probably have to basically soak my feet in it to worry. And the yarn, the textile itself after is not psychoactive, so you don't have to worry about wearing it either.
Very cool. Thank you.
Mm-hmm. So let's see. Oh, somebody
Someone was
asked.
asking about your social media.
Yeah. So probably most of what I do is actually on Facebook in various groups. I'm in like the Mushroom and Lichen Dyers United group. I'm an admin in a lot of the ID groups. I post some of my projects in like mushroom growing or mushroom breeding groups, and then just all over my own timeline. I do some on Instagram. I don't really like that platform as much, so it's mostly Facebook. And then not too long ago, I did start a Patreon where I like it a bit better than Facebook because I can post like really nitty gritty, long scientific documentation of what I've been doing. It's not formatted for photos as well. It's like, I've had mixed interpretations of the medium, but it has been a way for people to help me get stuff, which has been nice. I don't know. I still have mixed feelings on it. But it basically costs at least $5 per month. And then all of that goes to supplies, building my lab, etc. For people who can't pay, we do have a grant program because I hate the idea of information being behind any sort of barrier based on finances. So I've been letting people in through that route. But it's basically just where I post experiments. I recently have been doing chromatography on Cortinarius pigments. So Cortinarius cinnamomius. I've been doing TLC and HPLC on those, uploading the results. I recently had success dyeing with a new lichen. It's a new lichen dyeing method. The little red tips on some cladonia, incredibly potent naphthaquinone pigment. It can be handled basically the same as lobster and is probably one of the brightest purple reds I've seen out of any organism. The only problem is it takes a very long time to collect enough of the little tips. And it is a lichen that's difficult to collect ethically because it grows on the ground. But it was an amazing proof of concept, and that was really cool. That's up there. Some of the henna stuff, just kind of like random things I work on that I feel like are a bit too nitty gritty to put on Facebook. But a lot of it is on Facebook too. I guess I can, if anybody is interested in the Patreon at all, I can just paste it in the chat. It's moving mycology forward.
Please do.
There, that should have shared. Oh wait, that's a direct message. Okay,
Yeah,
there we go. There we are. And yeah, I mean, so it's 6 p.m. over here for me. I think we got most of the questions. Did anybody else have anything they were wondering about. Checking the chat.
I'm not seeing your information on
Well-
your Patreon in the chat yet.
Hmm,
Oh, me,
In
oh, there
my-
it is.
yeah.
Now I see it. Thank you.
Uh-huh. So let's see.
You know, if you can have that henna picture show up with the hands that you had
Oh,
from your
yeah,
class.
yeah. I can pull that up really quick. I just moved like a bunch of my photos from my phone onto a drive, some on my
Let's see if I-
computer. Let me see. Oh, yes, I do have it. Okay.
Oh, cool. 'Cause you got that kind of bluish color. I think you said it depends on how fresh it is.
Yeah, it depends on how fresh it is. It depends on the light you're using. And then I've noticed too, like different skin types can pick it up in different ways even. So like I've noticed some people tend to pick it up more yellow. Some people tend to pick it up more blue. So let's see.
Oh, you mean it varies
Skin to
from person to
skin.
person?
Yeah.
Ah,
Yeah, sweet. I can go ahead and share my screen again. Yeah, so if everybody can see that, this was after the IFFS class. This was art everybody had done on their own hands. This is my hand over here. I did 2 spores mating. This person was an actual henna artist. I'm sure you can tell. And then just like, I love seeing all the different designs. We got Mush Life up here. And this is something that's also really hard to photograph because like for one, my light is a bit of a spotlight. It shines mostly only one little area up. Like this is the brightest spot. And then like holding a light and a phone at the same time is kind of a nightmare. I've also learned how to mess with my phone to get better photos of it. Phones really hate fluorescent stuff, I've noticed. Like phones will often do a terrible job at photographing fluorescence. But yeah, this was after IFFS, just a great sampling of the art everybody did. Some of it like is so bright, it kind of washes out. Other stuff is like, there's all art going up a lot of these people's arms, but because the light's not big enough, you can't see quite all of it. But that's definitely one of the better photos I have of it.
You mentioned
awesome.
blues, and I was surprised you didn't mention Lactarius indigo.
Oh, well, that's because that one actually doesn't die, unfortunately.
Oh. Guys, my hand's nice and blue.
Yeah, so we probably need more people experimenting with that. Unfortunately, it's not a species I get in Oregon, so
We get them here quite a bit.
yeah, I can't really play with it. But one of the big problems is it is fugitive. So exposure to heat, like when you cook it, the pigment will degrade.
Right, yeah.
And it seems over time as well to oxidize and lose that blue color. The same thing goes on with like blue staining boletes as well, which you might think would be a great source. It oxidizes in basically every single species I've ever tested to brown, green, brown, yellow, or just vanish, like just nothing. I've never been able to get that one to stick around, but that is a group I'd like to experiment with more. There's just not really many blues. And there's one other blue that's worth mentioning. It's a bit more fugitive, but there's a lichen xanthoria. It's a really cool one. It actually dyes yarn pink or kind of reddish pink. But then if you leave that yarn in the sun, it turns blue. You can get kind of lighter blues from that. They're usually not outrageously saturated, but they are a little photosensitive, I've noticed. They kind of tend to shift more towards gray over time, but them and the tooth fungi are pretty much the only sources of blue that we have in the mushroom world.
Cool.
Yeah.
Well,
um,
thank you so much, Sydney. This was just amazing.
yeah, I'm glad it went smoothly.
Yeah, it did.
Yeah, doing 2 in a row. I mean, they're great subjects. I think I covered basically everything I wanted to, and the questions were great. I'm happy with the crowd and happy for you having me out.
And hopefully you don't think we overworked you.
No, no.
Okay.
Like I've said many times, I'll talk about fungi all day given the chance. It's definitely fun.
Yeah, somebody asked Cornelia once, do you think you could talk about fungi for 3 hours? And she said, do you think you can stop me at 3 hours?
That's the thing. It's like I scrolled back up and I see like a few more questions that didn't get addressed. It's like hard not to just keep going. It could never end basically.
Thank you
That's what-
so much.
Absolutely. Yeah, I had a blast. I was really glad when you reached out. And especially like sort of rolling off the fall season. Like I've had a lot of time to think and get talks together, and I just love giving them, really.
Excellent. Well, thank all the members who are still here, and we'll see a bunch of you on Saturday at the potluck, those of you who have signed up. And then Sunday is the Atlanta Mushroom Market at the Wild Heaven Brewery West End. And for those of you who went to that the first time, unfortunately, it seems like they're going to have music again. So it's could be loud, but they are putting our table. There's a speaker's room at the far end, other end of the brewery where I'm actually going to give a talk and some other people giving talks, but that's where MCG is going to have their booth set up. And so hopefully it will be much better. But if you're coming to the Atlanta Mushroom Market, consider bringing earplugs.
Right.
All right, excellent. Thank you, everyone. Thanks for hanging on. We went a little late, which was awesome. And
Sorry about that.
oh, not
apologize.
at all.
Um,
And lots of thanks in
Yeah.
the chat. So we and our club appreciate you, Sidney. And I'll be in touch tomorrow, probably by text about PayPal and stuff like that.
sounds great.
Okay.
Yeah, thank you. And thanks everybody for being here.
All right, thanks everybody. And
Thanks, Sidney.
and Ryan, give me an email by Friday night about whether you and Renee are going to be able to come or not. Okay.
Bye-bye.
I'll send you an email.
I appreciate that. Thank you.
All right.
Bye,
see you.
y'all.
Bye, everybody.
Well, that was fascinating. It was.